Hello, everyone. We're going to go over chapter 3, and it's a little bit long chapter, but it's okay. I think we can get through it in one session. It might not be your typical half an hour. It might be 40 minutes or so, or 35 minutes, but let's do the best we can. Okay. A cell theory, of course, it's a theory. It's not a fact. It's not a law, like a law of gravity. It's a law. It's a fact. But cell theory is the basic unit of life. Of course, I talked about it. All living organisms are made up of cells. Yes, that's true. New cells arise from pre-existing cells. Okay, so having said that, that's why this portion of the cell theory this is a fact. This portion of the cell theory, that's a fact, okay? This portion, it's making it a theory because we don't know where the very original cell came from, okay? That's up in the graph. Hopefully you guys become a PhD or very famous and you figure it out, but anyhow. So the cells come in different shape. For example, our nerve cells look like a star, and the neuron cells look like a spider, and then the red blood cells are round, but most generally in living organisms, the cells are round. Animal cells are round, and plant cells are rectangular, cubical, or something like that. Okay, cells size, it's important. You all know the size of the cells are very small. You have to look at it on a microscope, and then it has to do something with the surface to area ratio. And I'll let you read it. I don't want to spend too much time on it. It is important for cells to be small. That way you can have a total surface area of the cell. Look at it. The volume that this cell has, this block of cells compared to that cell can hold up. So it goes back to the surface area ratio, total surface area ratio, volume, and then of course, the total volume of all three of them, of all these, this one, and this one, they're all same if you look at the volume, but surface to area ratio, look at this one, it's greater. So it can hold up a greater amount of volume. Anyhow, what we look at cells on the microscopes, the compound microscopes, that's what we have at Delta College. And then I gave you instructions on YouTube videos when school was in session. It was part of mandatory. You take a quiz, or I call it oral quiz, that you can find and focus something on the microscope for me, an amoeba, paramecium, egg of starfish, anything you have. So, and I do have talked about it in the lab, if you go to the lab session. Here are the light microscope, if you would, and here's a transmission electron microscope. What happens, the difference between scanning electron microscope and transmission electron microscope, they cut the cell so you can see inside of the cell. With scanning electron microscope, you see a three-dimensional feeling. Still, it's a picture, but you get a 3D type of fixture, but you cannot look at the inside of the cell. It's much, much bigger than the view. The images are bigger than a light microscope, but the transmission electron microscope, they look at it, they break the cell down and you can see inside of the cell. Okay, these are the characteristics. I'm not going to spend too much time on these things. Okay, cells are classified on two groups, two categories. We already talked about that. I already had some quiz questions from it. So prokaryote and eukaryote. Prokaryotes are the cells that they do not have nucleus, and bacteria are in that group. Archaebacteria, eubacteria, they're all in that group. And then eukaryotic cells, in which we had 5 kingdoms. You remember the domain eukarya from chapter 1. There are 5 kingdoms: plants, fungi, protista, and Animalia, I'm sorry, four kingdom. We talked about that, and they are right here, okay, right in front of our eyes. And they are the eukaryotic cells, and they have nucleus, okay? So here is a transmission electron micrograph. They cut a cell inside of the cell, and you can see the nucleus right here, and you can see the endoplasmic reticulum, which I will talk about both of them. Nucleus of a cell is the brain of the cell, where all of the genetic material is stored. Remember, the prokaryotic do not have nucleus. They have genetic material, they have chromosomes, they have DNA, but they do not have a nuclear envelope, a nuclear membrane around it, and then, of course, all of the nuclear material, the DNA material is inside. Okay. Oh, this is in the lab. I showed you, if you look at the YouTube models, I took pictures, not YouTube, I'm sorry, the slides. I took pictures of the models we have in the lab, and these are the structures that you should be familiar with, and also the functions. Make sure you know the functions of this. There are not too many of them, 10, 15 of them. It's not much, 10 of them. And then you should know the structure, the functions. Both type of cells have plasma membrane. That's the commonality among prokaryote and eukaryote. They have cell membrane, I call it plasma membrane. And then of course they have cytoplasm, where all of the medium of inside of the cell, the medium right there, what is inside of the cell, that is the cytoplasm of the cell. And it's a semi-fluid, it's like a jello. If I extract it and without any other organelles, I put it in a dish, it's like a jello. It says, that's what they call it, semi-fluid organ, and the organelles are inside of it. Of course, the prokaryotic cells do not have organelles. It's only eukaryotes that they have organelles. The first cells on Earth were prokaryote, and there was also, these are all of the thing, and then endosymbiosis, it means the eukaryotic cells engulf a bacteria, archaebacteria, and then it became mitochondria, or they became a chloroplast. Here is the endosymbiosis evolution. The very first cells possibly had nucleus, but they did not have a chloroplast or mitochondria. And then here they are, they ingested a bacteria, and these bacteria became mitochondria, and also they ingested the bacteria that did photosynthesis, and then they became chloroplast in case of plants. Okay, plasma membrane, let's talk about the structure of cell membrane. Cell membrane, we talked about it already in the last chapter, chapter 2. It's made up of phospholipid molecule. It has a head and a tail, the two tails of fatty acids. The head is the phosphate group is in the head, and choline, and there are a few structures, of course, for you guys who don't mention. And a cell membrane has two layers of it, one here and one there. That's 2 layers of phospholipid. Right here, you can see one layer, like an Oreo cookies right here, and the cream would be in the middle. So this looks like Oreo cookies, if you would, right here, chocolate, and the white cream inside. And then there are protein molecules embedded, they're stuck right into middle of these phospholipid layers. These protein molecules, imagine them like a chocolate chip. So you put chocolate chip in Oreo cookies, that's what the structure of cell membrane is. So there are protein molecules. Attached to protein molecules could be oligosaccharides, sugar molecules, a few sugar molecules, these are all sugar molecules, or sugar molecules can be attached to the phospholipid. And that's what happens when you're blood type A, when you're blood type B, you're blood type AB, it's because of these sugar molecules right there, okay? Either a blood type O people do not have those specific sugar molecules. They have other sugar molecules, but they do not have those specific sugar molecules. Okay, plasma membrane continued at body temperature consists of a fluid mosaic model. So what is fluid mosaic model? It says that these protein molecules here, they do move around. these phospholipids, they do move around here. They're not fixated like the mosaic in your house. If you look at the tiles in your house, that's what mosaic means. They're fixated. They do not move around, okay? But that's not the case with these protein molecules. They do move around within the phospholipid bilayer. That's why they got the name fluid. Fluid, it means they do move around, mosaic model. Proteins freely move laterally. It contains cholesterol for support, and glycoproteins, that's what I was talking about. When there are carbohydrates attached to protein molecules, it's called glycoproteins. And when carbohydrates are attached to lipid molecules, they're called glycolipids. Okay, and then plasma membrane. Make sure you read all of these, you guys. Proteins act as a channel. They allow molecules to in and out. They allow water in and out. So they have a few functions. This protein molecule allowing water molecules get in and out of the cell without any expense of ATP. But it does not allow other molecules to get in and out of the cell. Large molecules, you know, they have to go, the macromolecules, they possibly cannot get through. They're too big. They cannot get through the cell. And then small non-charged molecules, they possibly can get in inside of the cell. They are not, they are not charged. They are not polar molecules. Okay. Then diffusion, osmosis, facilitated diffusion, active transport, endosymbiosis, and exocytosis. Quickly, I'm going to give you the definition of each, and then the next few PowerPoints is going to go over the details of it. Okay, diffusion is movement of molecules from high concentration to low concentration. There is no membrane involved. For example, if you have a beaker, you have a glass of water and you put sugar in there, so the sugar molecules, and you stir it, they're going to go from high concentration to low concentration. That's an example of a diffusion. You have a bottle of perfume, the molecules go from they come out from the perfume bottle and diffuses out through the room. So that's diffusion. Osmosis is movement of water molecules from high concentration to low concentration through a selective membrane, okay, through a selective membrane. So what I mean by selective membrane, that membrane almost has a brain. It can choose what goes in and what goes out. But the main difference between osmosis and diffusion, two things. One of them is a water molecule. Diffusion could be anything. It could be gas in gas. It could be solid in liquid. And the other one is selective membrane. So diffusion, in the definition, there is no membrane involved. Facilitated diffusion, well, when molecules go by osmosis, when molecules go across the membrane, if there is no ATP needed, that's called facilitated diffusion. If ATP is needed, it's called active transport. As simple as that. And then endosymbiosis and exosymbiosis, it's when large molecule, large amount of fluid or solid, what have you, get inside of the cell or get outside of the cell. That is called endosymbiosis, getting inside of the cell, and exocytosis, getting outside of the cell. Endo, it means inside. Cytosis, it means cell. Exo, it means outside, getting out of the cell. And cytosis, of course, it means cell. So these are, again, in your prefixes, suffixes. Diffusion, now he's going over the definition of diffusion, and he's talking about diffusion. I'll let you read it and get to the bottom of it, what diffusion is when the molecules go through when the molecules go from high concentration to low concentration. This is an example of osmosis because you have a membrane involved right here. So the water molecules are getting from high concentration to low concentration, just like that, okay? So, and osmosis, here it is, diffusion of water molecules from high to low water concentration. But again, they forgot to mention here, it has to go through a selective membrane, so through a membrane. And then there are three different types of osmosis: hypertonic. Hypo, it means low, and tonic, it means strength. Hyper, I've heard of somebody says he's hyper, is too much. Tonic, strength. And the other one is isotonic, same. Iso, it means same. So there are three different types of osmosis: isotonic, hypertonic, hypertonic. And if you place a cell, red blood cells, let's say it, or plant cells, you place it in a salt solution, what will happen? The cell will burst open. A lot of water goes, osmosis of solute, the cell shrinks. I'm sorry, I'm getting ahead of myself. This would be in case of salt solution. Sorry about that. This would be in case of deionized water, water, okay? So in case of deionized water, there is more water outside of the cell, and they go inside of the cell, they burst, the cell burst. This one, they call it crenation, when the cell shrinks. This one, the cell becomes huge and bursts open. Okay, here they are. Again, please read them and go over them for yourself. Facilitated diffusion, when molecules go in and out, there is no energy needed. No energy needed. And we talked about this in last chapter. When there is no energy, pretty much we are talking about no ATP needed. No ATP needed. Okay, this is a facilitated diffusion. It means when molecules go in and out, there is no ATP needed. Okay, active transport, it means ATP is needed right here. You do need ATP for molecules. You need energy for, in some cases, some molecules, not all molecules, they need to move the molecules in and outside of the cell. And it's an active transport. Right here, you're talking about sodium-potassium pump. This is called sodium-potassium pump. And then for that, I'm not going to go into details. There is ATP needed. Then, if you, as I said, big molecules are getting inside of the cell or outside of the cell, then you have endosymbiosis or exosymbiosis. Endosymbiosis, you have three types of them. One of them, type A, is phagocytosis. Phago, it means eating. And pinocytosis, it means cell drinking. And receptor-mediated endocytosis, it means there is a receptor that receives the molecules to allow them, and I'll show you a picture. So that's a third method of endosymbiosis. Here is examples of a bulk transport, and here is a receptor endosymbiosis. So this is a phagocytosis right here. This is pinocytosis, a lot of solute get inside of a vesicles. We call this a vesicle, and the vesicles pinch off from the cell membrane, and they get inside of the cell. And then for this vesicle have these receptors, and that's what they call it, receptor mediated. And then of course it pinches off. They all pinch off. They all get pinched off and they go inside of the cell. Or outside of exocytosis and get out of the cell. Exocytosis, transfer of molecule outside of the cell, and that's called exocytosis. Okay, now I'm going, the next few slides, it will talk about the organelles, the structures that is inside of the cell. So nucleus, it contains the instructions for genetic, for your genes, and sorry, I want to move this out of the way. I guess it's fixated there. So a nucleus, it contains the DNA material, and that is responsible for protein synthesis, making protein like your eye color, your skin color, your hair color. They're all distinguished by protein molecules. Endomembrane system, it means all of the membrane molecules, you know, the organelles. I did not give you this definition of organelle. I threw that at you before, but organelles are structures inside of the cell that have a metabolic function, and they're membrane bounded, like mitochondria. chloroplast, Golgi apparatus, rough endoplasmic reticulum, smooth endoplasmic reticulum. You had a picture of a cell with all of its organelles in a previous slides, 2-3 slides. So that's pretty much the best definition of organelle you're going to get anywhere. It's a membrane-bound structure inside of the cell that has a specific function. Usually it's a metabolic function, which prokaryotic cells do not have organelle. Okay, a nucleus contains chromatin, and then nucleoplasm, the fluid inside of the nucleus is called nucleoplasm, and not all cells have nucleolus. Some cells have nucleus, it's dark region inside of the nucleus, and it makes ribosomes. Here they are. So here is a nucleus, my pen. Here is a nucleus, nucleolus, nucleus right here, and outside of it is endoplasmic reticulum, and this is smooth endoplasmic reticulum. This is rough endoplasmic reticulum, smooth endoplasmic reticulum, which are two types of endoplasmic reticulum. Rough endoplasmic reticulum is responsible for protein synthesis. And then smooth endoplasmic reticulum has many functions. Lipid synthesis, one of them, lipid, and then the other one, detoxification in case of liver, detoxification in case of liver, and so on and so forth. So nuclear envelope, nuclear pores, nuclear membrane has these nuclear pores right here, so materials can get in and get out of the cell. Ribosomes, they're responsible for protein synthesis, and again, if there are so many ribosomes called polyribosomes. Endomembrane system consists of nuclear envelope, very important, you should know, endoplasmic reticulum, Golgi apparatus, lysosome, and vesicles. Each one of these, make sure you know the function of endoplasmic reticulum. You have two of them. Golgi apparatus, it packages cell material. Lysosome, it's a suicide bag of the cell. It's full of enzymes which can break down the product that is coming inside of the cell or they're leaving the cell. And vesicles are little packages that they come inside of the cell. You saw it in case of endosymbiosis. I'll show them to you. Rough endoplasmic reticulum, again, responsible for protein synthesis. Lipid synthesis has different functions, various cell types. Golgi apparatus, flatten, and it modifies proteins and lipids. It's a packaging plant. Green Bay Packers, anytime if you're a Green Bay Packers football fan, then Golgi apparatus should come to your mind. You should have no problem with Golgi apparatus, Green Bay Packers. They package cell material. Here, this diagram is showing you the endosymbiosis system that, the material from a nucleus can stimulate protein synthesis in rough endoplasmic reticulum, and those vesicles go to the Golgi apparatus, and then from Golgi apparatus, possibly they get modified maybe by lysosomes, and then the vesicles are formed and they get out, or the molecules that come inside of the cell, they go to lysosome break them down, and those particles go through Golgi apparatus, and then they get breakdown further or be modified. So I talked about all of these hydrolytic enzymes. The suicide bag of the cell, nickname. Nucleus is the brain of the cell. Lysosome is a suicide bag of the cell. Mitochondria is the power plant of the cell. Those are nicknames that scientists usually like to give it to these things. Cell junctions. So the cells are connected together. That's what we are going to talk about next, I guess. Cytoskeletons, cyto, as you know, as cell, skeleton is like your bones. It holds up the cell, okay? There is not much of it in plant cells because they have cell wall, but in animal cells, our cells, we have three different type of cytoskeletons: microtubules, intermediate filaments, and thin filaments. He's calling it, your textbook is calling it thin actin filaments. But anyhow, cytoskeletons, what we have, the microtubules, they are centrosomes involved, So please make sure you read all of these and know them. And if you have any question, holler at me. So cytoskeletons, actin filaments, intermediate filaments, cilia and flagella, as you know, they beat, so allow the organisms move. You have cilia in your upper respiratory tract. The only place we have flagella is human sperm. There is no other flagella in our body. Female uterus and oviduct have cilia. Our trachea has cilia, but nowhere else in the body we have flagella. Flagella is longer, flagellum is singular, of course, cilium is singular. And they both are inside of its microtubules, and they move things around. Here they are. It's called the structure 9+2. So they are actually, this is called as 1, 2, 3, 4, 5, 6, 7, 8, 9, and 2 in the center. So they call this 9+2 structure. And sperm has it, cilia has it, and it allows flagella or cilia to move. Extracellular matrix, stuff that is outside of the cell, and that is sometimes important in cancer patients. Collagen is an example of that. Elastin, these are all examples. Collagen, quickly, let's talk about this. Collagen is the most abundant protein molecules on planet Earth. So that would be a nice trivial question. So collagen is the most abundant protein molecules on planet Earth. And then I'll let you, fibronectin, these are the molecules that can be found outside of, and of course, I forgot to say inside of the cell too. So these protein molecules right here, outside and inside of the cells, they're called extracellular matrix. And they are attached to protein molecules within the phospholipid bilayer. You remember that? We talked about it. So 3 main types of junctions, the junctions between the cells, that's pretty much what it means. There are 3 different ways the cells are connected together. Adhesion junctions attach cytoskeletons of adjacent cells. Tight junctions produce a barrier. I hope you have pictures. If we not, I guess that's all. You have to know those. And gap junctions, 2 channels fuse, allowing communication between 2 cells. An example of a gap junctions in your heart. You have the heart, the cells in your heart are connected together by gap junctions. It's called intercalated discs. Okay, so intercalated discs are part of the, they are a type of gap junctions. Here we go, great. You have adhesions junctions right here. And then you have tight junctions. These are all protein molecules. This is phospholipid protein molecules. These are all protein molecules. And then in gap junction, there are channels, little channels between 2 cells that the molecules can go from one cell to the next cell. And that I gave you in case of heart. It's called intercalated disc, which in the lab, you look at it on the microscope. Okay, I think we, oh, no, we are not done. Metabolism, all chemical reactions that occur in the body, metabolic pathways, enzymes are responsible for metabolic pathways. So very important. What happens, a substrate that enzyme act upon. So you have a substrate and then you have enzyme right here, which it does not change, and you have a product at the end. So enzyme would be like a minister. You go to a church, you get married right here, then you would be a substrate, right? You go to a church, you get married, a priest is the enzyme, and you get out as a Mr. and Mrs. The other way around, if you want to get divorced, then you would be substrate, you and your spouse would be substrate. You go to a judge in a courthouse. The person who, the minister and the judge are not same. In some cities, very small towns, it could be same, but big cities, right now in Stockton, if you want to get divorced, you will not go to a minister. You go to a judge, you go to a court, and he will separate you guys, okay? So But at the end of the day, the minister, when you guys get married, he's probably single and being happy. At the end of the day, when you guys get divorced, then the judge is married and happy. He doesn't change his status. So the enzyme status does not change, but the substrate status changes to product. And of course, if you're going this way, you're going that way, the substrate status changes to product. Where in an enzyme, if you have an enzyme, big molecule, where the chemical reaction occurs right here, that is called the active site. It does not, the chemical reaction does not occur here, does not occur here, does not occur here, does not occur here, it occurs here. So that portion of that enzyme is called active site. Here they are. This diagram shows everything that I talked about. Make sure you study them. Make sure you know them. Lowers the activation energy. It means if you have an enzyme, you do not need so much energy to separate molecules or combine them together. That's pretty much what it means. Coenzymes are sometimes vitamins and cofactors. They did not mention it here. It could be minerals, cofactors. They are minerals, calcium, that they help enzymes to proceed their functions. Here is saying this is a chemical reaction occurring without the enzyme. This is much energy you need. This is a chemical reaction, same chemical reaction occurring with the enzyme. So this much energy you don't need. That's pretty much what it means. Mitochondria, as I said, is a power plant of the cell. ATP, the glucose molecule, the meat, the fat you eat, eventually it turns to ATP. Okay, so that gives you energy. Of course, it goes through cellular respiration, and I'm not going to talk about it in great detail. When you take Bio 1, we talk about it. Here's a mitochondria, look like a footprint. This is a transmission electron of mitochondria right here. This is a transmission. Of course, this is a drawing of a mitochondria. This is a real picture right here. This is a real picture, and this is just a drawing. Okay, ATP production, adenosine phosphate. We talked about all of these, glycolysis, citric acid cycle. Make sure you study these things and you know a few things about them. If you have any question, let me know what happens inside of here's a blood vessels when glucose goes inside of the cell, breaks down to 2, 3 carbon molecule. These 3 carbon molecules, I put the covalent bond between them, it's called pyruvate. Let's try pyruvate. Okay, that pyruvate molecules goes into mitochondria and it breaks down eventually all of these carbons. All of these carbons end up as carbon dioxide. Okay, pretty much. That's in a nutshell. And then when, of course, that sugar molecule end up into carbon dioxide, you get a lot of ATP from it. I hope I'm making some sense. Okay, so that ATP allows you to think, allows you to run, allows you to take an exam, take a quiz, move, and so on and so forth. Okay, the chemical reaction that occurs in the cytoplasm is called glycolysis. You do not need oxygen for it. But the chemical reactions occurs here in mitochondria, you do need oxygen for it. And that's what they call this aerobic respiration here in mitochondria. When sugar molecules break down to 2 molecules of pyruvate, it's called anaerobic respiration. Anaerobic respiration. Okay, so I gave you a nutshell what that is, and I hope you studied it a little bit more so you can answer the quiz and exam questions. If you have any question, let me know in the Zoom meetings. I can help you a little bit. Okay, so that's pretty much what I talked about. Electron transport chain, fermentation is anaerobic respiration, and that's the end of the material for this. All right.